Today our PowerPoint will cover the topic of enzymes, so make sure you have the notes, enzymes, and something to write with. As we move through this PowerPoint, I will be going through each number on those notes, and I will give you the answers to fill in. Proteins are made of amino acids. So question 1 in your notes, what monomer are proteins made from? And your answer, number 1, would be amino acids. So here on the slide you can see different pictures of amino acids. They're represented by different colors where I'm circling, or just the 3-letter abbreviation that means them. So like alanine and arginine and aspartame and cytosine or cysteine, leucine, and so on. But as that long chain of amino acids come together, which are called proteins or polypeptides, they're going to start to fold up into secondary structures, which look like these alpha helices, and eventually into beta sheets. And then they're going to fold a little bit more. Again, all these proteins coming together. You can see the alpha helices and the beta sheets within the tertiary structure, so just folding up until it eventually hits its quaternary structure. And so you just need to recognize this picture as a protein as well, this like jumbled up blob, which is just the quaternary folds of these amino acids here. So to recognize an amino acid, remember on one side you should see nitrogen, and on the other side you should see a carboxyl group. So really when you're looking for proteins, look for the NCCO groups. So look for the NCCs to recognize a protein. And here you can see it says R group. That R group is just the variant piece of each amino acid. So the reason why all of these amino acids are different over here, why they're different colors and why they have different names, is just because that R group makes them different. And proteins can have carbon, hydrogen, oxygen, nitrogen, and sulfur. So you might see CHONs, carbon, hydrogen, oxygen, nitrogen, and sulfur. And so it's just some extra information. It's not actually something that's written on your notes, but you might be asked on the star. Small proteins are called polypeptides. Poly meaning many, and then peptides. Question 2, what is another name for proteins? And you're going to write that green word in there, polypeptides. The reason why it has this name is because of the bond between the amino acids is called a peptide bond. So here we see LYS, which is lysine, and VAL, which is valine, and that bond that's holding them together is called the peptide bond. Again, here's another picture of an amino acid structure. So you just need to recognize proteins as having that nitrogen and then that CCO group. So amine and carboxyl group on either side. And again, that R group is just what makes all of these amino acids different because they're made of different elements. But only carbon, hydrogen, oxygen, nitrogen, and sulfur. So again, CHONs. Enzymes are one of the many kinds of proteins that are made by cells. Enzymes are found in every cell, and they speed up chemical reactions inside the cell. So question 3, enzymes are a form of what biomolecule? You're going to say proteins. They're a special type of protein. And question 4, what do enzymes do? And what's written in the yellow is what you're writing. They speed up chemical reactions. They speed up chemical reactions. It does say that they're found in every cell, but also note that there are thousands of different types of enzymes in every cell because they all do a different thing for our body. Every chemical reaction requires energy to get it started. that energy is known as the activation energy, and sometimes it's abbreviated E with a little a, activation energy. Question 5, define activation energy, and it's just the energy to get a reaction started. So energy to get the reaction started. So in the graph to the left, here is where it started. But in order for this reaction to happen, I need this much energy. So I need to get this much energy for this reaction to start. So that's my activation energy. So enzymes speed up chemical reactions, specifically by lowering the activation energy. Again, that's the energy required to get a reaction started. So enzymes are just going to go and look at the graph below. They start in the same place, but they're just going to lower the activation energy. So it's a lot less energy required to start the reaction. So maybe this is like 100 joules, where without the enzyme, that red graph here, you would need maybe like 1,000 joules. I like to think of it as before you're going to go and start a run, before you go and start a marathon, a big long run, you don't want to go up a mountain to get to your run. You'd rather walk up a hill. It's just less energy that you have to use before you go on your marathon run. So energy here is a lot less. So that reaction won't happen a lot faster, where this one's going to be a lot longer. Okay, so enzymes speed up reactions, as you can see by the reaction rate here on the X, by lowering that activation energy so that reaction happens a lot faster. So question 6, how are enzymes able to speed up chemical reactions? Make sure you wrote by lowering the activation energy. Enzymes speed up chemical reactions without being changed. So we know enzymes speed it up, but now we also know that they're not going to be changed. That means enzymes are reusable. And another name for that term is called a catalyst. So a catalyst is something that speeds up chemical reactions without being changed. They're reusable. So down here is a picture of an enzyme. And right here is its active site, and that's where the substrate, which is in green, can bind to the enzyme, and it's the same shape. So if it wasn't the same shape, this substrate could not bind. Okay, when it's all together, it's called the enzyme substrate complex. No other substrate can come in at that time, it's already bound. So now that reaction is going to get sped up, and the products are made. So you can see how they changed from a green color to like a blue and an orange color. And those products, they go and get released. So we started with one substrate, and it got broken down into 2 products. So that's an example of a catabolic reaction. Because it got broken down. Remember, anabolics to build. So if I had 2 substrates coming in and it came out as one product, that would be anabolic. So enzymes can be used over and over again by the cell. Now this enzyme, you see how it's not changed? It stayed exactly the same as when it started. So now the next substrate can come in and do the same thing. So question 7, how are enzymes affected by the reactions they direct? They are not changed. They stay the same. So I would say they are not changed. And then question 8, how many times can a single enzyme be used? And you can just say over and over again, because it's really indefinitely, that enzyme always stays the same. So we just keep using it and keep using it. Any substrate that speeds up a chemical reaction without being permanently changed is called a catalyst. That's what I just told you on that last slide. So for question 9, define catalyst. say it's a substance that speeds up a reaction without it being changed. Again, it's reusable. Remember, it's reusable. There are many different catalysts used by chemists in a lab, but cells always use enzymes. So again, here's that graph. The red dotted line is with an enzyme. It speeds it up by lowering the activation energy, so less energy is required to start that reaction, so it happens a lot faster. Question 10, what kind of catalyst do cells always use? And you want to go and write enzymes. The reactants, or the raw materials that enzymes start with, are called the enzyme's substrate. So here's the substrate. And remember, that has to be a certain shape. It has to be the same shape of the active site on the enzyme. Question 11, what do we call the reactant that attaches to the enzyme? And you want to write what's in green, substrate. And then question 12 says, label the diagram shown below with the following terms. So you need to go and write substrate and point it to this yellow object, the circle diamond shape. And then point enzyme at the Pac-Man shape. And then the Pac-Man's mouth. Label that as the active site. And this is on your paper for question 12. When the substrate actually binds to the enzyme's active site, that's called the enzyme substrate complex. So you need to go and label that for the second blob that's right after the first arrow. And then after the last arrow, again, go ahead and label your enzyme, which is your Pac-Man. And then show me that these are your products. So the substrate's attached to the enzyme at its active site, and then the enzyme goes and directs that reaction. 412, make sure that you write that this is an example of a catabolic reaction, because it went and took the substrate and it broke it down into two products. So catabolic is to break down. RXN is reaction. So I'd go ahead and write this is an example of a catabolic reaction. In question 13, the substrate always attaches to what part of the enzyme? And you want to say here at the bottom in green, the active site. Because the active site is designed to attach to a very specific substrate, cells must have different enzymes for every chemical reaction that happens in our body. So we have thousands of different enzymes in our body, so they can all do different chemical reactions. Question 14, why does a cell need a different enzyme for every reaction? Because the active site is, the substrate's very specific to that shape of the active site. So say the substrate is very specific to the shape of the active site. If it's not the same shape, like this picture here, it will not bind. So that's why we have thousands of different enzymes that have different shapes of their active site. And then even the smallest cells have over 1,000 different enzymes inside. So question 15, how many different enzymes are in even the smallest cells? And you want to say over 1,000. So for this part of the notes, I want you to go back to Google Classroom, and attach to where you found this presentation is a video link. There might actually be two. I want you to watch that video, or if I did put two, watch both of those videos, and then come back and finish the few questions that are on the back. I think you only have 2 more questions. So watch that video. It's really short on enzymes and how they work. And if there's 2, watch both. But don't forget to come back and finish the last 2 questions. Alright, so to help you remember what an enzyme does, we have this little meme for you, and he's called the crime-fighting catalyst. Remember, catalysts speed up chemical reactions without being permanently changed, so they're reusable. So we have something like the enzyme, it's like the flash, and he's speeding up your metabolism 1 chemical reaction at a time. So he's making those things faster, whether it's metabolizing, you're breaking it down, or building things like an anabolic reaction from 1 or 2 substrates into 1 product. So question 16, what is the enzyme's nickname? And you want to call him the crime-fighting catalyst. And question 17, what is his superpower? I want you to say speeding up your metabolism 1 reaction at a time. Please look at the board and see what your next directions are, or raise your hand so I can give you your next task.